begin with, the first instrument to be talked about is the Secchi disc. The Secchi disc is a water quality parameter check that was created in 1865 by Angelo Secchi to measure the water transparency, or in other terms, the turbidity of the water. It comes in the shapes of a circular disc with black and white color divided into 4 quarters with similarity to the concepts of yin and yang. The size is standardized to 2 different sizes, 30 cm and 20 cm in diameter, since its invention. The disc is usually mounted by a pole line and lowered into the water until a depth at which the disc is unable to be seen anymore.
To use the Secchi, first the Secchi was lowered into the water slowly on the shady side of the boat until it is not longer visible. Then the depth was recorded. Each of the type is 1 meter. Then the disc was then raised until it's become visible once again. Then the depth was recorded. Used to measure the water clarity, where can help to monitor the changes of water parameters. It is playing a role to measure the transparency of water to determine the amounts of plankton.
Compound light microscope. The compound light microscope is one of the famous microscopes commonly used in the laboratory to observe magnified image of cells and other microorganisms. The compound light microscope is a tool containing 2 lenses which magnify and a variety of knobs used to move and focus the specimen. It has high magnification of microscope, then use 2 lengths to compare the level of magnification. The first length is referred to the objective length and have 4 power, 10 power, 40 power and 100 power magnification. The second length is referred to the eyepiece length. This length compound or multiply the magnification of objective length by another 10 power, resulting in a total magnification of 40 power, 100 power and 400 power and 1000 power.
First, switch on the microscope. And here is the light source to control the light intensity of the image. The image will see through eyepiece, and we will start from the lowest magnification, which is 4 times magnification. The cost adjustments is used to adjust the stage, either it will be lift or lower down. And here is just fine adjustment where used to focus and sharpen the image. After that, we may change to higher magnifications. Do not adjust cost adjustment knob. Only fine adjustment knob and light source will be adjusted to make the image clearer. It is functions to explore and investigate the morphology and size of the plankton. It's important to observe the microorganisms which can't be seen by naked eyes where inhibits on our Earth.
Sediment sieve. The sediment sieve is used to determine the particle by size within the sample. It has many mesh sizes to determine different sediment by filtering out the material. Sediment sieve have 1 mm and 500 micrometer size that we see at the laboratory. This equipment can be used on non-organic or organic grain material, including sand, crushed rock, clay, grain, and seed, depend on the method used. The result of particle sizing from this equipment help to identify the sediment layer.
This is an equipment known as sediment sieve, which used to filter the sediments. First, we may put the sample on the sieve and use water to rise through the sample. The water will flow out from the bottom of the sediment sieve. After that, the sediment sieve can be separated and we can see the contaminants that left over on the lower sieve. It is used to sort out invertebrate sample from the sediment sample. To talk about the important, sediment sieve is used to study the invertebrate sample which can be found in the sediment.